From 2014 to 2016, thousands of people in West Africa became infected with the Ebola virus. This was the largest outbreak ever recorded, causing a significant loss of life and profound social and economic disruption in the region. After cases emerged in the United States and Europe, the virus also raised a question of our own preparedness to epidemics like this and how we can prevent such outbreaks from ever occurring on a major scale in the first place. The Ebola virus disease is fatal in nearly half of all cases, and in order to prevent another outbreak, we need to create a vaccine as soon as possible for all 4 disease-causing Ebola variants.
The Ebola virus is a ribovirus, which means that it uses RNA-dependent polymerase for replication. The virus, which is a string-like structure around 1,000 nanometers long and 100 nanometers in diameter, is composed of a membrane, Ebola glycoproteins, matrix proteins, and a nucleocapsid. The Ebola virus genome only encodes for 7 proteins, all of which are structural. The glycoproteins studded on the outermost layer of Ebola virus particles are capable of changing their shape and therefore their ability to bind to various types of cells in the body. While there aren't many cases of the virus as compared to other infectious diseases, such as cholera or HIV, it's still extremely deadly and contagious. We've been lucky to only have experienced a few epidemics, and we've gotten through past ones with the efforts of global and local health authorities. But given the erratic nature of these outbreaks, we have to be prepared for the next Ebola epidemic.
The Ebola virus is transmitted from animals to humans and between humans through bodily fluids such as blood, vomit, sweat, urine and feces, or sexual fluids that enters our body through mucous membranes, scratches, or from a needle. Ebola targets immune cells such as macrophages, monocytes, and dendritic cells, endothelial cells, and liver cells. These cells and natural killer cells aren't infected but also start to die off. Ebola virus, being surrounded by glycoproteins that bind to receptors on these cells, trick the host cell into engulfing them. Brought into the cells in lysosomes, and once the lysosome is at the right position in the cell, the lysosome membrane and the viral membrane fuse and the virus releases its genetic content. Ebola virus is extremely effective at crippling one's immune response. Macrophages, which are infected by Ebola, uncontrollably release cytokines and other molecules, causing several blood clots in the bloodstream and triggering a massive inflammatory response that leads to a severe hemorrhagic fever, better known as the Ebola virus disease.
Ebola uses the lytic cycle for replication. Genetic content from the virus, in this case RNA, uses the host cell's machinery to create replicas of itself. This replication is at the expense of the host cell. Eventually, this causes the host cell to rupture or lyse, which spreads replicas of the virus further. As the virus continues to spread and be replicated, cytokine levels drastically increase and a hyperinflammatory response is sustained, eventually leading to internal bleeding, organ failure, and often death. So we know how the virus infects our bodies. What can we expect from a patient suffering from Ebola? The incubation period for the virus lasts from 2 to 21 days, but symptoms will most likely appear in roughly a week. The immediate symptoms are fever, chills, muscle pain, fatigue, and a sore throat. After some time, the symptoms become much more severe. Patients suffer from severe diarrhea and nausea, and from seizures, flaking skin, and rashes. They also experience skin hemorrhages and bleeding from the eyes, ears, nose, and mouth. With these deadly symptoms, people usually die between a week or 2 weeks. Very few people survive, but those that do have red blood cells that can release proteins to fix damaged blood vessels. Additionally, survivors tend to have a temporary inflammation response.
There are 6 distinct strains of the Ebola virus, each named after the location they were discovered in.
The Sudan Ebola virus is a member of the filovirus family. It was introduced as a strain of Ebola virus in 1977 after the 1976 Sudan outbreak. Although no reservoir host has been identified yet, scientists believe bats harbored the virus due to its distant relationship to the Marburg virus, which has been isolated from bats. The Sudan Ebola virus is also uncharacterized on the molecular level, but scientists do know its proteins perform similarly to the proteins in EBOV.
The Bundibugyo Ebola virus originated in Uganda, is also a member of the Filoviridae family. The first case of VDBV appeared on August 1, 2007, and the presence of Ebola virus was confirmed on November 29, 2007. The outbreak was declared over in February of 2008 until a second outbreak broke out in August of 2012. Ebola virus disease in humans due to BDBV cannot be differentiated from Ebola virus due to any of the Ebola viruses by clinical observation alone. Just like Sudan Ebola virus, BDBV does not have an identified reservoir host and is believed to be harbored by bats due to its distant relationship to the Marburg virus and Raven virus. It also has proteins that function similar to the proteins in Ebola virus.
The Thai forest Ebola virus originated from a single human infection from touching a chimpanzee in the Thai forest. The contact occurred when a scientist conducted an autopsy on a chimpanzee, which is believed to have consumed a western red colobus monkey, causing the infection. This was the only Ebola virus that had one infection. The infection was not fatal, and the patient had recovered in 6 weeks. It's also a member of the Filoviridae family and was introduced as a new strain of Ebola virus in 1995. The Thai forest Ebola virus was the first time a human Ebola virus had emerged outside of the Congo River basin, but its location is not surprising due to its environmental similarities to the tropical rainforests in the Congo River basin.
Bombali Ebola virus was first reported on July 27, 2018. Scientists trapped the bats in Guinea and Liberia in 2018 and 2019. Found traces of Bombali Ebola virus RNA by using reverse transcription PCR in kidney and lung samples acquired from 2 M. condylurus bats in Yalanjo village and in liver and spleen tissues of N. condylurus bats from a school in Nzoroke. They also took blood, intestine, and brain samples, but they were negative for any viral RNA. After conducting many tests, researchers believe that Bombali virus may not be pathogenic in humans, but they are not sure yet. They did conclude that the Bombali virus is more closely aligned with the human pathogenic viruses than Restin, which is not human pathogenic, but they believe it might be due to the origin of the virus.
The Restin Ebola virus came from Virginia. Scientists conducted an experiment on 4 piglets and found that the piglets infected with Restin Ebola virus developed severe respiratory tract disease and shed the virus from their upper respiratory tract. Although Restin Ebola virus cannot affect humans, it's known to cause the disease. The experiments the scientists conducted brought up concerns that pigs could be a bridge for the virus or an amplifying host. However, the Restin Ebola virus was first identified in 1989 when research monkeys from the Philippines were shipped to Reston, Virginia, and a second outbreak occurred in the Philippines, spreading between the pigs. The outbreak was the start of a pig-to-human RESTV transmission, so the World Health Organization put out a global alert in February of 2009. Scientists have also identified RESTV sequences in pigs in China and are monitoring the pigs for disease throughout Southeast Asia and the Philippines.
The cause of the first Ebola pandemic in 1976 in Zaire, now the Democratic Republic of the Congo, and the 2014 and 2016 West African epidemic, Zaire Ebola virus has the highest fatality rate. The Zaire Ebola virus was thought to be a strain of the Marburg virus. It received its own distinction in 2010. Reservoir host is assumed to be fruit bats and is transmitted between humans and the fruit bats via bodily fluids.
The West African Ebola virus epidemic, the most extensive outbreak of the disease in history, has had a massive cultural impact on several nations in West Africa. Treatment and containment of individuals carrying the disease have been an integral component of curbing Ebola outbreaks, but this often comes into conflict with local cultural norms and practices.
Several West African communities, the tradition of standing by the ill, that is, being physically present around the sick or injured in your community and helping to nurse them back to health, has been around for centuries, and it is meant to show respect and to express solidarity with the patient. Custom was severely restricted by Ebola isolation procedures and often caused friction between communities and Ebola treatment centers, where Ebola patients would be quarantined and monitored away from their families.
Especially in rural areas, traditional medicine and witch doctors take precedence over Western medicine because of their ease of access, affordable cost, and strong public trust in them. Several West African cultures associate illnesses with etiologies unrecognized by Western medicine, such as the influence of ancestral spirits, affliction by gods, or spiritual possession. Ritual and herbal treatments are often believed to be an effective medical treatment, and having been around for several generations, gained the trust of many communities.
safe burial of those who have passed away from Ebola virus is also a very controversial issue. Traditional funerals are often major cultural events with ceremonies that can last for multiple days. They often involve touching the deceased, both in the preparation leading up to the funeral and during the ceremony itself. After the conclusion of the funeral, the body is buried on land adjacent to the deceased individual's home.
Burial rites are extremely significant to families who believe these traditions are necessary to honor the dead and to ensure their spirit will feel happy and not forgotten. Interference with funerals are viewed as not only incredibly offensive to the spirit of the deceased, but capable of putting the family in grave danger. Improper burials are believed to inflict the family with illness and harm in the future. Medical workers have to be very sensitive with handling these sort of situations and need to address these issues with understanding and compassion. But often carrying out their duties is inherently at odds with local traditions.
There's also a generalized resistance to health authorities in several regions. In parts of Africa where armed conflict, bureaucratic infighting, and corruption is endemic, a lot of people have lost faith in their governments. Sometimes citizens view foreign medical aid as a front for corrupt governments to receive international sympathy and handouts from other nations at the expense of the livelihood of Africans, despite it being a lifeline to the areas hardest hit by Ebola virus epidemics.
Overall, given the deadly and contagious nature of the virus and the widespread perception of Ebola treatment and containment as invasive, vaccination is a critical step towards eradicating the virus. Here's how a vaccine would work. Innocuous virus particles in the vaccine would be spiked with proteins from the Ebola virus. This modified virus isn't able to cause any harm in the body, but our immune systems are able to recognize the Ebola protein. This would prompt an immune response where our B lymphocytes would produce antibodies against the Ebola protein. Therefore, if an individual comes into contact with the actual Ebola virus, they have a very good chance of fighting it off because their immune system should now be capable of producing antibodies against the virus.
However, vaccines require a huge amount of capital before production even starts. Vaccine R&D can go into the hundreds of millions, and potential vaccine candidates need to go through several clinical trials and periods of review by the Food and Drug Administration before being approved. Mass production, storage, and distribution of the vaccine also requires their own infrastructure, which comes with its own costs. Ultimately, we believe there is a lot of value behind creating a vaccine for the Ebola virus. It might just be what prevents the next pandemic.